So this is lab 17, and this is going to have a kind of quick lab today on k-means clustering. And of course, this lab is lab 17 associated with lecture 17, which was also on k-means. Okay. And so we want to look at k-means, and of course we have our favorite data set, which is our iris data set, right? So we can grab our favorite friend iris, right? Sepal length width, petal length width, and we have species. Now, this is maybe not the best example to use for clustering because it does have a species, but we could see, if you didn't actually have the species, maybe we could remove that species variable, and we could see can we kind of recover that species in a kind of unsupervised way, right? Now, in reality, in clustering, if you wouldn't have something you're trying to recover, but it's a nice example because we know there's some underlying true structure there, the name of the species, and we could see if that's recoverable using k-means. So let's take our data and let's just remove out which of these columns it gives us species. So if we just remove that, now instead of having 5 variables, we have 4, and we just have kind of the x's. So this is just our x's. And we can try applying k-means to this, and there is a function here called k-means, which is just built into the base r. So k-means is a really classic algorithm. It's been around a long time, and so it's basically built into the base R. You don't need to load any packages. There's nothing complicated. It's been around forever, and so it's just base implemented in R. And this is our k-means clustering. You give it your x, either a numeric data matrix or something like that. You can also there also are k-means versions where you can just give it dissimilarities, and we will look at that. Not this lab, but you can also have one where you just give dissimilarities. There are different algorithms. Here's Lloyd, or Lloyd's algorithm. A couple of these things, the number of iterations allowed, so the number of centers. So you can, one way that people get around the problem of the random initialization, because you could randomly initialize your thing and get different solutions, is if you just do a bunch of random initializations and you take the best one. So that's one way people get around the problem that this isn't guaranteed to give us the globally optimum solution. If you set it to randomly initialize a certain number of times, then you just take the best clustering as measured by within cluster sum of squares. And there's some other arguments here, but the main ones are those. And it will return to us a bunch of stuff: the clustering assignments, the centers, i.e. the means, total sum of squares within sum of squares for each cluster, etc., between sum of squares, etc. It gives us, so it's a really nice little algorithm. So let's see if we can do it. Let's say km.out, or k-means output. We can run it on k-means, give it my data, my x. We know there are 3 centers, so let's just go for it, right? So centers is number, it's k, basically. We know there are 3 true groups. A realistic situation you wouldn't really know, and we can talk about that a little bit. And then let's just do one initial start, and let's actually use Lloyd's algorithm. Super quick. Because it doesn't have to test all possible subsets, right? We know that our matrix has quite a few, I think it has, what did it say, 150 rows, so that would be really complicated to calculate all possible cluster assignments, but it doesn't. It uses some kind of greedy approach to try to approximate that solution. So if we just print it, it will give us some information on the k-means clustering. So it finds sizes of 50, 62, and 38. We know it should be 50, 50, 50 because we know we have 3. We know the true, quote, true clusters. We know the underlying structure of the species, which are evenly split. Nice thing about unsupervised learning is there's no real test train split. You don't have to deal with that. You don't have to deal with cross validation. Blah blah blah. At least not in as serious of a way. And it gives you some the cluster means. So because we have 4 variables, our cluster means are kind of four element vectors. And it gives my cluster assignments within cluster sum of squares. And so it gives us between over total, which is basically something like an R squared. It's basically the amount of, so sum of squares here you could is what's used for numeric data, where you have squared Euclidean distance. In the lecture we had called it kind of the total dissimilarity. In this case, it's the dissimilarity is a square, and we sum it up, so it's a total sum of squares. So the between sum of squares and the total sum of squares is basically like R squared. It's basically how much of the variance, or the sum of squares, is explained by the clustering assignment. So it says here about 90% or so, 88% is explained by the clustering assignment. So that's pretty cool. So it seems like these things cluster pretty well. So we can get our cluster assignments as that gives us our vector of cluster assignments. There are 150 corresponding to 150 rows of our data matrix. Now, if you remember, our data is 4-dimensional. So we run into a slight issue because I'd like to plot these clusters, right? But I can't plot a 4-dimensional plot. Luckily, we already know a dimensionality reduction technique that I can use for visualization. I could basically do PCA on my data and use the first 2 principal components, project my data down to the optimal 2-dimensional subspace, and at least it would allow me to visualize it. Even though it's not exactly what the data is, it lives in the 4-dimensional subspace, we could actually just plot it, project it down to the 2-dimensional subspace. And the way I'm going to do this is, I'm not even going to use, you could use PR comp or whatever, but principal components analysis is basically just the SVD, so I can just do the SVD of my data. And then I could, you could get the weights and you could multiply the data by your weights, but we know that that is basically just the U matrix. And so the first 2 principal components are basically the first 2 columns of that U matrix. Now, I haven't scaled my data, I haven't centered it. I guess I could scale it if you want to be more proper. Okay, and, basically my first 2 principal components are basically the columns of U. They're actually stretched according to the variances, but I don't really care for the purpose of visualization. Who cares, right? So if I just plot this, right? So this is a whole digression, right? I'm just basically using PCA to visualize my data. So here is my data projected down into 2 dimensions. And we could have done this way back when we talked about classification. We could have done this instead of just picking 2 of the variables, which is what I did there. I just subset 2 of them and I plotted it because I said, I don't want to deal with this. But the kind of big boy way of doing this, and the way you do this, a better way of doing this, is to find the best 2-dimensional subspace, and plot that 2-dimensional subspace, which is basically what I've done here. So, so that's kind of data projected down using PCA or the SVD or whatever you want to think about it, projected down to that 2-dimensional subspace. And then I can, of course, so first of all, my X labels, my X-axis is basically PC1, my Y-axis is basically the second component. And I could make my colors for this plot. I could grab the cluster assignments, and I could color the points in my plot by the cluster assignment. And if I do that, I get something like this. Voila, a gorgeous picture. And it's basically recovering the three species in my data. How do I know that? Well, I don't know. There's a function called text, which, how do I do this? I think first we plot. Do we plot first? There's a function called text. Let's see if I can do this correctly, which will allow us to not just to actually add, instead of having circles here in my plot, as my plot symbol, I can add text. And so what I'm going to actually add is if I add as my label, I can add the species, the actual species. Let's see if it does this correct. I don't think it's going to allow me. Okay, let's do this. I think first you have to plot it. This is why I shouldn't do anything on the fly. And then I could add text. Yes, so I add text, X, Y, text color, and the label. Okay. Here we go. So the color is the group assignment, and the text is the actual true, quote, true species. So the Setosa, right, it's clustering pretty well, and the Versicolor and Virginica are closer, so it's, the clustering assignment is more muddled. So you see most of the Versicolor are red. I guess I shouldn't, can I not make, no, it doesn't matter. The color actually is also the true, because I set it to be the true color here. In any case, you can basically see that the color is the assignment, sorry. So you can see that the Versicolor is basically red, so that red cluster is basically the Versicolor, and the green cluster is basically the Virginica. And you probably can see some that are kind of not perfectly correct. If we're to zoom in here, yeah, so you see like a red Virginica and a green Virginica. Okay, so the clustering, I mean, again, typically in a real clustering, you don't really have a metric to say good or not, but since we actually know the true underlying structure, that's what we get. And so of course, I could play this game. We chose 3 in this case because we know that there are 3 true clusters, but we could try this with other numbers too and see what we get, right? So for example, really to my eye, if I didn't know, I would say there's 2 clusters, this one over here and this one over here. So there's kind of the Setosa and other cluster. So I could have said, maybe it's 2, and this is the clustering assignment we would get. Of course, you can play this game with other clustering assignments, maybe 5, and probably I should really stop playing with the parameters. Probably just let it. There's some other fancier versions of algorithms beyond Lloyd's try and solve some of these issues, some other issues that Lloyd's might have. So let's just leave the algorithm default, and you might want to try, say, I don't know, 5 random initializations and take the best of those 5. So you could put n star and say 5 in that way. So here is our clustering with 5 centers. The number of centers is not related to the number of starts here. So this could just be 10. It doesn't really matter. Here's my clustering with 5 clusters. Yeah, okay, it doesn't seem to make a lot of sense, but in a real problem, you wouldn't know the actual kind of true underlying structure here. So that's probably a good place to stop. That's kind of the basic of application of a k-means clustering and how we apply it.